Welcome to the second video in the unit on microbial control. In this video, we're going to switch and start talking about antimicrobial drugs. So we're going to look at the different properties that those antimicrobial drugs have and kind of define what an antibiotic is. Let's go ahead and get started. In the first video, we talked about how different chemical and physical methods could be used to control microbial growth. However, keep in mind, those are all done on surfaces. So even antiseptics that are used on living tissue aren't things that you use internally in the body to remove microbes from. They're only used on the surface of living tissue. So what that means is if you have a microbial infection in the body, we're going to need to use something else. Instead of using chemical antiseptics and disinfectants, we need to actually use drugs that are meant to go inside the human body. And the idea here is that we don't want to damage the human tissue also. So we're looking for drugs that will harm and kill the microbe, but not harm and kill our cells. Your chemical antiseptics and disinfectants don't do this because they don't have a specific target. They're usually going to harm lots of molecules in the cell. So for example, ethanol will dissolve lipids and denature proteins. Your cells have lipids and proteins too. Things like chlorine will oxidize molecules like your DNA, just like eukaryotic cells have. So that's why we don't want to use those within the human body. Instead, we're going to use antimicrobials, which are chemotherapeutic agents, they're drugs. I want you to keep note of the terminology. An antimicrobial is a drug that can be used against a microbe. That's kind of a broad umbrella term for lots of different classes of drugs. Antibiotics are used only against bacteria. So the targets that they're aiming for are found in bacterial cells, but not in other microbes and not in your cells. Antivirals we use against your viruses. Antifungals we use against fungal pathogens. And then antiparasitics we use against parasites. We're mainly going to focus on antibiotics in this unit, but we will talk a little bit about antivirals and some antifungals and antiparasitics towards the end. As we get into the discussion of antibiotics, I want to highlight two things. The first is that before antibiotics, infectious diseases were much more serious and they caused much more fatal disease. So for example, 90% of children that were suffering bacterial meningitis died because we didn't have a way to treat that, and 80% of wounds infected with Staph aureus was fatal. In fact, people would die from wounds that they got from stabbing themselves with a sewing needle or puncturing their skin with a rose thorn, just because that small little prick was all you needed to get Staph aureus into the bloodstream, and there were no ways to treat it. Before antibiotics, childbirth was also much more dangerous, as up to 40% of new mothers actually died from childbed fever. So antibiotics have likely extended our life expectancy by about 8 years by now being able to treat these infectious diseases. But the other thing I want you to notice is that this chart here shows the death rate for infectious diseases in the United States from the 1900s up to 2000. And what you should notice is that there's a pretty steady decline in the number of deaths from infectious diseases well before penicillin was first introduced. So penicillin was used here, first used in 1942, and you notice death from infectious diseases had been decreasing. And that's mainly because of public health measures. So you can see we've added chlorine to water here. We started setting up health departments. A lot of this was based on sanitation, so cleaning up things like raw sewage. And all of those help to lower the death rate. And the thing to notice, this big gigantic peak here in number of deaths in that 1 year, that's the 1918 flu, which you've actually been hearing more about in the news because that was the last really big pandemic that we had that highlighted a large number of deaths worldwide. So antibiotics are defined as a chemical that's produced from one organism that can kill or inhibit the growth of a bacterial organism. So it's important to note these are natural products that are actually produced by microbes in nature. Humans have just taken them from the microbial world and used them for our own purposes as drugs, but they're really ways for microbes to have chemical warfare on each other. A lot of soil microbes produce antibiotics because there are lots and lots of microbes in 1 gram of soil. And so these allow ways for one organism or one species to make an antibiotic, kill off its neighbors, and be able to get more food source and have more area to live in. Natural antibiotics are produced by molds and some bacterial species for this kind of chemical warfare purpose. We now also have what are called semi-synthetic antibiotics, where we as humans have taken those natural antibiotics produced by the microbes and chemically modified them in a lab. And so this is doing chemistry in a test tube, and what it does is it allows the antibiotics to have kind of increased properties, maybe be able to fight more organisms. Some of our antibiotics are still natural antibiotics, others are what we consider semi-synthetic now. All antibiotics are what we call selectively toxic. That means they really should be able to fight and kill bacteria, but not harm our own human cells. They also show what's called a spectrum of activity. Not all antibiotics are going to work against all bacteria, so we need to look at the mode of action and what types of organisms the antibiotic can actually interact with and kill, and we'll talk about that as we go through this lecture. The first antibiotic was discovered in 1929 by Alexander Fleming. And I really like the story of how Alexander Fleming discovered penicillin because it highlights that not everything in science happens as planned. Sometimes you just have to be paying attention at the right time. So Alexander Fleming studied Staphylococcus aureus, the organism that causes staph infections. And back in the early 1900s, staph infections were much more serious than they are now. In fact, the majority of people died from staph infections, from even very small wounds, because the bacteria got in the bloodstream and there was no way to stop it from growing. So Alexander Fleming had accidentally left one of his Petri plates with Staph aureus growing open on his desk when he went on vacation. And when he came back, he realized that there was something besides just Staph aureus growing on the plate. He saw this mold colony that you can see here in this picture. And the other thing he noticed was not just, oh, his plate got contaminated, but that around that mold, there seemed to be a clearing. We call that now the zone of inhibition. But what Fleming thought was, maybe this mold is producing something that seems to be killing or stopping the growth of the Staph aureus on that plate. And it would have diffused away from the mold into the plate. Turns out he was right. And so Fleming worked with this mold, which is now called Penicillium notatum, and recognized that it was producing a molecule that killed Staph aureus. Except Alexander Fleming wasn't a chemist, so he wasn't able to purify penicillin. That took the work of another team called Howard Florey and Ernest Chain, who actually figured out how to purify and how to grow enough Penicillium notatum that you made enough penicillin to actually purify and use as a drug. They finished this production in 1942, and if you know anything about the 1940s, this is really the start of World War II. And so there was a large push at the time for these guys to really develop this production chain because World War I, more soldiers had actually died of gangrene, which is caused by staph, than actually died in battle. And so having something that could kill staph and prevent gangrene in soldiers was a huge boom for World War II. All three of these men were awarded the Nobel Prize in 1945 for the discovery of penicillin. A year later, we actually then figured out the chemical structure of penicillin itself. A researcher called Dorothy Hodgkins did this, and she went on to win the Nobel Prize for that in 1964. So a lot of Nobel Prizes associated with penicillin, and the structure was important because now it lets us make semi-synthetic antibiotics by taking that chemical structure and doing chemistry in test tubes to modify the structure slightly. Just like there were things to consider when choosing the right chemical or physical agent to control growth, there are also a number of things you have to consider to prescribe the right antibiotic. The first is that antibiotics, just like chemicals, can be both bactericidal or bacteriostatic, which means they might kill the bacteria or they might just inhibit its growth. If you have a bacteriostatic antibiotic, in the body, it stops it from growing, which gives your immune system time to get into gear and go off and kill the microbe itself. However, if you're immunocompromised, bacteriostatic antibiotics aren't necessarily the best choice because you might stop them from growing, but your immune system might not be able to go off and kill them on their own. In that case, you probably need a bactericidal antibiotic. Other things you have to consider is the spectrum of activity. So what types of organisms does the antibiotic actually attack and kill? What is the right dose? And so we'll talk about what a chemotherapeutic index is and why that's important. Is the pathogen you actually have an infection with susceptible to the antibiotic, or has it developed some sort of antibiotic resistance? And then the last one is, are there any synergistic or antagonistic effects with the antibiotic and other drugs some patient might be taking? And this is something you have to consider for any drug that you might prescribe. You always need to consider how it's going to interact with other things the person is taking. For example, there are some antibiotics that actually interfere with birth control pills. So if you're using birth control to prevent pregnancy, you sure want to know that to make sure that you're using something else while you're on that antibiotic. We're going to talk about these things that you have to consider in the next couple slides. The spectrum of activity of an antibiotic describes the types of organisms that that antibiotic is effective against. And it's important to note that some antibiotics are effective against a large number of organisms. Those are called broad spectrum antibiotics, and you'll often hear them abbreviated as broad spec. Or there are some antibiotics that only function against a narrow range of microorganisms. We can see some examples of those here, and you don't need to memorize this table, but it's just a good way to highlight. Things like penicillin G are really only effective against a small number of gram-positive cocci. That's different than something like ciprofloxacin, which you can see spans gram-negative cocci, gram-negative bacilli, or things like cefabopime, which scans all the way from gram-positive to gram-negative bacilli and cocci. So something like ciprofloxacin would be considered more broad spectrum. Something like penicillin is considered more narrow spectrum. And it's important to know the organism that you're trying to treat to determine what antibiotic to use. If you don't know the organism you're trying to treat, so if you haven't identified the pathogen that's causing disease, you often will pick a broad spec antibiotic because that increases your chances of the antibiotic actually functioning. The downside of that is that broad spec antibiotics, because they kill or interfere with large numbers of microbes, are also going to destroy much of your own normal flora and the bacteria that are keeping you healthy. This is why a lot of times you'll have gastrointestinal problems if you're on an antibiotic. It's because you're messing with the normal flora of your GI tract. And so if you do know the organism you're trying to kill, it's much better to use a narrow range antibiotic that's more going to target just the pathogen and won't harm your microbiota quite as much. When deciding to use an antibiotic, it's not just about what antibiotic to use, but also how much of the antibiotic to give. So what dosage should you be prescribing, and how should it be prescribed? So in the table at the top, you can see the different routes of administration and what that does to the concentration of the drug in the blood. We can see if we do an IV administration, we get a very peak high in the bloodstream. This is typically used if you have severe sepsis or bloodstream infections because you want to get that antibiotic concentration really high right into the bloodstream. The other two ways are intramuscular or oral, both of which have a lower peak, but you can see they last a little bit longer in the bloodstream itself. A lot of this is important because as you look at that concentration, so remember we've got increasing amounts in the blood, and we've got time. What happens based on the route of administration is that we're going to increase the amount, and then eventually that decreases down. This decrease is because there's a half-life of all drugs in the body. Your body is actually metabolizing them and removing them from the bloodstream. And what will happen is there's some specific dose, so there's some specific amount that is the MIC of the drug. So we have to stay above that minimum inhibitory concentration, or else the antibiotic is not going to be successful in the body. And so what happens is here we're above the MIC. As time goes on, we drop back below the MIC. And when that happens, now we need a second dose. This is why typically when you're prescribed antibiotics, you take one in the morning and one at night, because your one in the morning increases the concentration in the body above the MIC, and then eventually when you fall below that concentration, you take another dose, so you lift that amount back up above the MIC to ensure that the antibiotic is actually functioning. If the concentration is below that MIC, it doesn't matter that you're taking the antibiotic because it won't be at a high enough dose to actually kill the microorganisms. Another property to consider is the chemotherapeutic index of the drug itself. All drugs have a chemotherapeutic index, so this isn't unique to antibiotics, but it's a key feature of antibiotics because they have a very high chemotherapeutic index. What this means is that they're very safe drugs. They don't have very high side effects because they're typically not harming our own cells. Remember, antibiotics are usually going to have targets that are unique to bacterial cells, so they tend to not have a lot of side effects and kill our own cells. What the chemotherapeutic index is we're looking at the concentration of a drug, so the concentration of the antibiotic given, and the percent of population that's actually affected. And there's going to be two curves on this. So as the concentration of drug that you give increases, eventually you're going to hit a concentration that it starts to work. So this is treating the disease. So if this is an antibiotic, this is the concentration that we need to be above that MIC and actually prevent infection from happening. And so we'll see that goes up and eventually will plateau and affect all the people. What we care about in this is going to be the 50% mark. And so if we come over at 50% and we come down, whatever concentration this is called the effective dose or the ED50. It's saying what concentration of drug effectively treats this pathogen in half of the population. Now, if you keep giving higher and higher concentrations of the drug, eventually you will start to have toxic side effects. And so now at a higher concentration, we're going to have at 50% some amount of drug, which we call the TD50 or the toxic dose 50. This is when we're now starting to see side effects from the drug itself. What your chemotherapeutic index is the ratio of the toxic dose divided by the effective dose. So for example, if this concentration was 100 and this concentration was 1, our chemotherapeutic index would be 100. The higher the chemotherapeutic index, the lower the side effects. Because what this is saying is you can give any concentration between here and the drug is going to be effective and it's not going to be toxic. If we drew one of these curves for something like chemotherapy drugs for cancer, what you end up with is an effective curve, say here, and then your toxic curve almost right on top of it. And this is because cancer cells are technically your own cells, which means most of the time your chemotherapy drugs that are killing cancer cells are also in some way harming your own cells, and this is also why you see lots of toxic side effects to those drugs. We're going to come back and talk about the chemotherapeutic index when we also talk about things like antivirals and antifungals. So it's important you understand this concept and the fact that the selective toxicity of antibiotics gives them a very high chemotherapeutic index. They're toxic for bacteria, but not for your own cells. One of the considerations when deciding what antibiotic to use is whether or not your pathogen is susceptible or resistant to the antibiotic itself. To figure this out, you have to do antibiotic effectiveness testing or figure out what is the MIC, which is the lowest concentration of the drug that's needed to stop the growth of the pathogen. This can be measured 3 ways: broth dilutions, e-tests, or Kirby-Bauer discs. The broth dilutions are done as shown in this image. So you use increasing amounts of whatever your antimicrobial agent is. So this first tube doesn't have any antibiotic in it, and then you increase the concentration of antibiotic as you go in each one of your tubes. All of the tubes are then inoculated with your pathogen of interest, and you looked at the turbidity of the tubes, so where is growth happening? So we can see here we've got growth in all of these tubes, even though you can tell by the color that the growth is getting less. And then here we have no growth, and we have no growth in this one. So the very first tube that shows no growth tells us what the MIC is. So if we went lower than that concentration, we did not inhibit the growth, but if we went higher than it, we saw no growth. And this is what those tubes look like in real life. You can see the turbidity of the bacteria growing, but those tubes there are still clear. One interesting part with broth dilutions is they also let you test the MBC, or the mammalian bactericidal concentration. So the key with the MIC is it tells us the lowest concentration that stops growth, but it doesn't tell us whether that is being bactericidal or bacteriostatic. We don't know whether the bacteria in tube 6 are dead or if they're just not growing. And so after doing the broth dilution test, what you can do is you actually take a small sample out of this tube and you're going to plate it onto a Petri plate. You do the same thing with tube 7 and we look for growth. So if we see colonies growing on this plate, what that tells us is that in tube 6, the bacteria weren't dead, they just weren't growing. If we took them out and put them on a plate with no antibiotic, now they can grow just fine. But tube 7, we saw no growth in, so that tells us tube 7 is the minimum bactericidal concentration, the amount that will kill the bacteria. But tube 6 is just the MIC, and it's inhibiting the growth. While the broth dilution tubes allow you to calculate the MBC, they typically are no longer done in clinical diagnostic labs, mainly because they're cumbersome and they're time-consuming to set up all those tubes, and they take a little while. Instead, people typically use either the Kirby-Bauer test or the E-test. So in the Kirby-Bauer test, you take each one of these little paper discs that are infused with antibiotic, and you put them on top of this Petri plate. The plate itself is covered in what we call a lawn of bacteria, and what we're looking for is the zone of inhibition, so the clearings that we see around each one of those discs. The size of that zone of inhibition is going to determine the susceptibility of the organism. The bigger the zone, the more susceptible, because the more effect that had and the more it prevented the growth. In order to measure this, you actually measure the diameter of that clearing, so of that zone of inhibition. And you can see here the E antibiotic for this species seems to work pretty well. We have a big zone of inhibition there, suggesting that that species is susceptible to the antibiotic and we could use it for treatment. But in this plate, the E antibiotic has a very small diameter, almost no zone of inhibition at all, which suggests that this species is resistant to the antibiotic. The antibiotic is not able to inhibit its growth. E-tests are a little bit different. So the Kirby-Bauer test, you compare to standards and it can just tell you, is this susceptible or is it resistant? So should you use the antibiotic or should you not? But the E-test will actually let you calculate the actual MIC, so the actual concentration of drug that meets the MIC. And that's because we use, instead of discs with a set amount, we use a strip that's embedded with different amounts of antibiotic. So you can see at the top we've got 256 micrograms per mil there, and way down at the bottom we have 0.016 micrograms per mil. And so you get a dilution of antibiotic all the way down the strip. Up here at the high concentrations we have a big zone of inhibition, and then as the concentration decreases, we're seeing less and less inhibition until we hit a molecule here where this would be the MIC, because if we go below this concentration, we no longer see any zone of inhibition. Things below that concentration are not able to stop the growth. And so you can use an E-test to actually calculate the MIC of whatever species you're working with.